Hello, everyone, and a really big welcome to our webinar, Mastering Food Chain Microbiomes. My name is Paul Cotter, and I'm the coordinator of the Master Project. And we're here for approximately the next 2 hours or so to hear about the exciting results and activities from this project, which has been running for over the course of the last 4 years or so. Just to let you know in advance that the sessions are being recorded, just in case you enjoyed so much you'd like to listen over and over again. And the sessions today will be chaired by Lisa Mahler de Sanchez from QIAGEN and Tanya Kostic from the Austrian Institute of Technology, both of whom were integral members of the project, as well as myself. So in session one, our researchers will give an overview of the research outputs from the project. Session two will present some of the project's innovations. And in session three, we'll have a panel discussion addressing life beyond master. So the future of microbiome-related technologies and their applications to the food chain. We have a large master team who are available online today to answer your questions at the end of each session. If you do have questions, please type them into the Q&A box at the bottom of your screen, and if possible, the name of the person that you'd like the question to be directed to. We really would love to hear from you. If we don't get through all of the questions today, don't worry, we'll do our best to contact you after the webinar with some answers. So let's get started. So before we go any further, I want to tell you just what exactly the MASTER project is. So MASTER is an acronym for Microbiome Applications for Sustainable Food Systems Through Technologies and Enterprise. And it's important for you to know just exactly what a microbiome is, I guess. So a microbiome relates to any environment in which you find microbes. The combination of the different microbes you find in that community, so bacteria, fungi, viruses, and so on, are known as the microbiota. And the various other components within that environment, such as the DNA within those microbes, the proteins, the lipids, the toxins they produce, and the environmental conditions and so on, are referred to as the microbiome. So understanding microbiomes can provide meaningful individual and public health solutions, impact on climate change, and help transform our food systems. However, for very many years, microbiome research focused predominantly on human health aspects, and the tools and new technologies that were being developed hadn't really been applied to the food chain and food industries and consumers. So to address this, the European Commission a number of years ago decided to fund 5 large projects relating to food systems microbiomes with a focus on food innovations, food consumers, food production. So MASTER was one of those, together with 3 sister projects known as Circles, Holo Food, and Simba. And these were pulled together under an umbrella referred to as a coordination and support action. And this was known as microbiome support, led by Angela Sesic. Now, MASTER and the sister projects are innovation actions, which means that the research carried out needs to be of direct relevance to consumers and to the food industry by the end of the project or soon thereafter. So not blue skies fundamental research, but really meaningful research that can impact on people's lives almost immediately. And we and the other partner projects have been working very closely on a number of joint initiatives, including an approach whereby at Microbiomes for Life, you can find out about different aspects of microbiome research. And these tools and resources have been developed specifically for the public. So I'd encourage you to take a look at this website. Moving on and talking specifically about MASTER. So what was the focus of MASTER? So what we have done is to take a global approach to the development of microbiome products, foods and feeds, services, and or processes with high commercial potential. And now, rather than focusing on just one food system, we set ourselves the very high goal and target of looking at improving the quantity, quality, and safety of foods across multiple food chains, spanning the marine, plant and soil, rumen, meat, brewing, fruit, vegetable waste, and fermented food-related industries. In order to achieve this really considerable target, we worked together with another 28 partners from right across the EU. These included several different researchers from universities, from other research performing institutes, from companies, both large and SMEs, and other different entities to achieve this. And to have achieved those very considerable goals during the last 4.5 years was really remarkable. especially considering the fact that while we did get to meet occasionally, the vast majority of progress due to the COVID pandemic needed to be online. And despite that, we ended up being considerably successful. So as I mentioned, we're focusing on a number of different environments. And within those different environments, we've been developing new strains and consortia, so individual microbes or groups of microorganisms, new services and processes, new foods and feeds, new molecular analysis, and new databases and pipelines. And over the course of today's webinar, you're going to be hearing some stories relating to individual outcomes and impacts from those different research initiatives. But before getting into those, I'm going to provide some broad brushstroke overviews relating to just exactly what we were doing. More specifically, Master had 4 different work packages in which we carried out different types of research. Work package 2, where we focus on plant, soil, marine microbiomes, 3 for the rumen, 4 for food processing, valorization of waste streams and human gut health, and work package 5 focused on integrating new microbiome technologies for the food chain. The first of these, plant and soil microbiomes, what we did was to look at new microbiome solutions for improving traits of other crops. And this included improving nutrient uptake and providing control of pests and different microorganisms that are pathogens and can cause problems for our fodder producers. We also looked to develop new microbiome-based diagnostic tools to detect those pathogens and also to study the soil to predict how well they would respond to different types of interventions. From a marine microbiome point of view, the focus was to develop new sustainable feeds for enhanced fish growth, and also to employ, again, new microbiome-based diagnostics to assess fish health and to detect pathogens before they become problematic. The rumen space was one that we paid particular attention to, given its important impact on society with regards to sustainability and the control of things like methane and other greenhouse gases. And in this regard, we have made considerable inroads with regards to utilizing host genomics and breeding to alter the rumen microbiome in a beneficial way and to also impact the animal phenotype, so the animal features. We've defined the rumen microbiome at different stages of life in animals to improve efficiency, reduce methane production, and to improve the quality of the milk and meat that's produced, and also to develop new tools and models that we can predict how an animal will respond to different interventions. Work package 4 was another very large endeavor and spanned a number of different areas. One of the most ambitious was to carry out a microbiome analysis or mapping of food processing facilities right across Europe. In essence, we collected thousands of samples from hundreds of food processing facilities, but rather than use old agar-based traditional microbiology testing, we pushed the boundaries and used DNA sequencing-based approaches to study those microbiomes and develop new tools and approaches that will be relevant to industry going forward and mean that these new technologies will become relevant very soon. We also look to optimize fermented food microbes and processes, improve biopreservation, develop innovative functional foods, and to prepare and develop minimally processed foods that are designed with a view to enhancing gut health. The final research work package is work package 5, where we carried out an integration of microbiome technologies. And in this regard, we've been developing, optimizing, standardizing, and validating a range of different microbiome analysis, which in the future can be employed to identify rapidly spoilage and pathogenic microbes within food processing facilities to enhance the safety and the shelf life of those foods. We developed new computational pipelines that facilitate the rapid analysis that's required in order to get the quick results to employ those new technologies, and also to develop new long-term resources that will be not just of relevance now and in 6 months' time, but continue to be of value to people right across the food chain. So now that we've set the scene, let's continue and launch into session 1. So session 1 focuses on the research outputs from Master. Now, we were very successful, and there's too many to summarize adequately in the time available today. So we picked 7 presentations in particular to focus on, corresponding to 7 different research outputs. These presentations are short snapshots, so maybe 3 or 4 minutes long, I hope you enjoy them, and we'll return to a question and answer session corresponding to those questions or corresponding to those presentations in a little while. We'll now start those presentations.
Hello, my name is Fiona Brennan. I'm a soil microbiologist in Teagasc and Johnson Castle. And today I'm going to give a quick summary of some of the research we've been doing investigating whether microbial bioinoculants can be used as biofertilizers in grasslands. So why are we interested in grasslands? Firstly, grasslands constitute about 70% of all agricultural land. And in countries like Ireland, this can be up to about 90% of all agricultural land, making it a very important land use. All food systems need to be both economically and environmentally sustainable, leading towards a push towards reducing chemical fertilizer in these systems, which can be both costly for the farmer and also can be associated with negative environmental impacts on water quality and on air quality, including greenhouse gas emissions associated with fertilizer application. So finding alternative methods of providing nutrients to plants and supporting growth of plants can be very advantageous on multiple fronts. So biofertilization strains are one technology or one option that could be used to replace these fertilizers. And one of the groups that's often used for this purpose is arbuscular mycorrhiza fungi, or AMF for short. These are fungi that form symbiotic relationships with plants. And in return from carbon from the plants, they provide a range of nutrients to the plants derived from their extensive networks, mycelial networks below ground. In this project, we used 3 of these strains together. We also looked at another group of organisms, plant growth promoting rhizobacteria, and these were able to mineralize nutrients and improve plant uptake. So we looked at 2 bacterial strains, a bacillus strain and a Parabarcoldaria strain. These isolates were used in tandem and also in singly. We looked at how these strains did in both monocultures of perennial ryegrass and also more diverse sward types where we incorporated legumes and herbs. Our main questions were, did these strains colonize? Do they persist long enough to have an impact? What's the impact on the quality and quantity of the herbage? And what's the impact of soil fertility in terms of whether these strains work or not? We're also interested in the impact on the wider plant and soil microbiome. Our key take-home messages were, yes, we could get successful colonization of these strains, and they did persist. We found in terms of yield, sward composition had the biggest effect on yield, but for some consortiums, so for example, our AMF applied in concert with the bacillus, we did get increased uptake of calcium, of nitrogen and phosphorus, and trends of increased yield. We found, or we learned, that compatibility really matters in the formulation of these consortia, and some organisms work much better together than others. Also, the soil fertility context is really important in terms of whether these organisms will work or not. So just finally, to acknowledge my colleagues in Teagasc, particularly Israel Ecoy, who did most of this work, and our partners in AIT, Christian Hansen and Enoch, and our funders for the Horizon 2020. Thank you very much.
My name is Rennie Groben. I'm a project manager at the microbiology group at Martis, and I will give you a short presentation about the research on increasing bioavailability of sustainable aquafeeds and food safety. With the growing global population and the need for healthy, nutritious food, seafood is in demand as never before. However, current fishing practices put a lot of pressure on marine ecosystems, and fish stocks worldwide are often in danger of overfishing, and this makes this practice unsustainable in the long run. Aquaculture, which is the rearing of aquatic organisms in large tanks on land or in open pens in the ocean or in lakes, has the potential to solve this problem and produce high quality food in large amounts. As a result, aquaculture quickly became the fastest growing industry in the food sector. There are, though, problems associated with growing aquatic organisms in aquaculture, and one of them is the need for sustainable sourced food and feed. At the moment, the necessary protein sources and largest component of aquaculture feed is fish meal, meaning that low-value fish is caught in the ocean or reared and to feed the more valuable fish in the aquaculture. This system does, of course, not solve the problem of overfishing and depletion of fish stocks, and in the long run, this is also not sustainable. So we need to find alternatives. In Master, we did trials with different kinds of alternative protein sources in aquaculture feed for Arctic char and Atlantic salmon. We used proteins from waste streams of the dairy and beverage industry, and as well as insect meal, and replaced a certain percentage of the fish meal in the feed with it. In addition, we supplemented the feed with selected prebiotics and potentially probiotic bacterial strains to test if they increase the bioavailability of these feed. We think that these might change the composition of the microorganisms in the fish gut and have beneficial effects. We measured the impact of these treatments by analyzing the growth rates of the fish, the quality and sensory values of the fish fillet, the gut microbiomes, and the product safety through microbial and chemical analysis. The results showed that fish meal could be replaced by up to 15% with alternative protein sources under prebiotic supplementation without any negative effects. There was even an indication that some of the probiotic bacterial strains had growth-enhancing properties both for Atlantic salmon and Arctic char when they were administered with the feed. Furthermore, the studies showed that these probiotics inhibited the growth of pathogenic listeria on fish fillets without having any adverse effects on the quality of the fillet. So this makes them a valuable tool to improve food safety in the aquaculture. We think that these beneficial effects could have been achieved through modulation, meaning changes of the microbiome, the microbial composition in the fish gut. To summarize, our research has been shown to the possibility to use alternative and sustainably produced protein sources in aquaculture in combination with pre and probiotics. Not only have they no negative effects, but we could even show that they might increase growth and reduce infections with the pathogenic listeria, making them a promising way forward towards safe and sustainable aquaculture production. This work was done within the Master project by researchers from Martis and Ifremer, and we would like to thank all the colleagues for their successful combination.
Oh, hello, everyone. I'm David Yáñez, a researcher working at the Spanish Research Council in Granada, Spain. And I'm going to quickly tell you some of the results we obtained combining rumen enteric methane inhibitors with hydrogen acceptors. We know that ruminants such as cattle, goats, and sheep, they establish a very strong relationship with a complex microbial ecosystem that degrades part of the plant material that come into the diet, and in that degradation activity, they produce different compounds, one of them being different volatile fatty acids, acetate, propionate, or butyrate, that represent the main source of energy for the ruminant. In that kind of fermentation, also hydrogen is produced, and methanogenic archaea in the rumen are responsible for using that hydrogen to produce methane, so the fermentation can continue. The production of this gas, methane, which is a potent greenhouse gas, contributes significantly to the emissions of greenhouse gas emissions of the animal agriculture. So different strategies have been developed, one of them being the use of methane inhibitors that inhibit the activity of methanogenic archaea to reduce the production of methane. When these methanogenic archaea inhibitors are used, there is also an excess of hydrogen produced in the rumen that is wasted because it's not used to produce energy yielding compounds. Different alternatives have been proposed in the last years to try to capture this excess of hydrogen, so the animal can not only reduce the production of methane, but also obtain a benefit from the metabolic point of view. Some of these initiatives include the use of acetogens, sulfate reducers, and nitrate reducers, but there are some limitations associated with these strategies. We are proposing a different strategy using different phenolic compounds that are present in plant materials, that when they are degraded in the rumen, they produce volatile fatty acids that can further contribute to the energy production for the animal. So to test this hypothesis, we ran a different set of in vitro trials combining a potent methane inhibitor like Asparagus taxiformis, a red algae, with a range of different phenolic compounds, as you can see here, to see what the effect was on rumen fermentation. And we could see that when asparagus was used, there was a significant reduction of methane production that was also associated with a huge increase in hydrogen production, as you can see here. When Asparagopsis was combined with these phenolic compounds, in some cases, this excess of hydrogen was reduced. And in the case of fluoroglucinol, one of the phenolic compounds, there was an increase of total volatile fatty acids. So meaning that this hydrogen in excess was used basically to produce compounds that the animal can use as a source of energy. And this is basically as a result of fluoroglucinol being degraded in the rumen to produce acetate, and in that process uses hydrogen, and in this case, part of the hydrogen that is generated in excess when methane is inhibited. We had a chance to further test and confirm this in vivo trials in goats in respirometry chambers, as you can see in here in this photo. Again, using a controlled diet compared to either Asparagus taxiformis, fluoroglucinol, or the combination of the methane inhibitor with the phenolic compound. And we could see again that Asparagus taxiformis significantly reduced methane. But when we combined the use of Asparagopsis with fluoroglucinol, there was not only a reduction of methane, but also an increase in total volatile fatty acids, confirming what we already obtained in vitro. So the conclusions are that combining a potent methane inhibitor such as Asparagus taxiformis with a phenolic compound that uses the excess of hydrogen represent an attractive combination for farmers to reduce methane emission, but also provide more energy for the animal. Thank you very much for your attention.
Hello, everybody. My name is Vincenzo Valentino. I am a postdoctoral researcher from the University of Naples Federico II, and the title of my presentation is Novel Strategies for Microbiome Mapping in the Food Industry. Basically, we know that in the food industry, microorganisms are present on surfaces during the food processing. However, even though we clean the surfaces, we know that a bunch of microorganisms are still present on these surfaces. Basically, the microbial loads resisting on surfaces after the cleaning and disinfection is quite limited. So they are difficult to be studied using current methodologies. So we wondered, who is there, what can they do, and also why and how do they survive? To answer these questions, we applied the standard operating procedures developed within the master project, which are based on detection and study of the microbial DNA. To answer this question, we test the procedures in two industrial settings. One produced the minimally processed vegetables, while another one produced ice creams. What we did was basically collecting environmental samples after the cleaning and disinfection, collection of ingredients and products from the first production following the environmental sampling, and then in the laboratory, processing of samples, detection of DNA, and data analysis according to the standard operating procedures developed. So here are the main outcomes from these studies. We identified the communities residing on clean surfaces, highlighting that they might be very complex, so made of lots of species. This is a very interesting result that was impossible to achieve using the current methodologies that are used to study the microorganisms associated with the food industry. Also, we detected microorganisms genes involved in production of biofilms. Biofilms are thin layers strongly attached to surfaces, embedding and protecting many microorganisms from disinfectants. We also, our results also are in accordance with the previous evidence, which suggests that the presence of these biofilms might also lead to selection of superbugs that are microorganisms that show antimicrobial resistance to several compounds. Finally, we also predicted that a great proportion of resistant microbes living and selected on clean surfaces might be transferred to the final product. And this might might arise some problems regarding the quality of food products and also for safety reasons. So overall, the advantages of the standard operating procedures that we test and the future outcomes are here presented. Basically, we were able to quickly detect spoilage and even pathogenic bacteria. And so these procedures might be useful in the future to reduce food waste and also to prevent foodborne outbreaks. Also, we were able to achieve a higher resolution in profiling microbial communities and their potential compared with traditional methods. So overall, in the future, stakeholders might soon implement these standard operating procedures as a routine practice to monitor microbial communities residing in their industries. This may be very useful to develop ad hoc sanitation strategies that are tailored towards some specific microorganisms that need to be eradicated from the specific industry. So thank you for your time.
Hello, my name is Ines and I'm a PhD student at Teagasc, and today I'm going to talking about the development of probiotic ingredients from apple waste. The cider sector in Asturias produces around 12,000 tons of apple pomace per year that is used normally as a compost for animal feed or just waste. We have focused our work on apple varieties under PDO Cider of Asturias, and we selected 9 varieties based on their resistance and production. First, we made a general characterization with these 9 varieties, and we saw all of them has a high mineral content, high fiber, and galacturonic acid content. Because that, we made a pectin extraction with these 9 varieties. The raw material was freeze-dry, green, and low the pH with a citric acid at 2.5. Then we made a conventional citric acid extraction, and we obtained around 10% of the yield. With the results, we selected these 3 varieties, Solarina, Regona, and Perico, because they are interesting pectins due to their structural difference. With these 3 varieties selected, we made another 3 pectin extraction methods, the same conventional method but at a low temperature, and 2 more methods with ultrasound, bath, and probe modes. The yield was not improved, but we obtained pectins with different structural characteristics that can improve the fermentative properties. Based on these results, and with these 3 varieties selected, we made a batch fecal fermentation. First, we made a simulated digestion with all these substrates, and then fecal fermentation with fecal samples from IBD and healthy donors. We made this fecal fermentation with medium and 0.5% of substrates, and we take samples at different times for taxonomy sequencing. The results of this part, we saw a statistical significant increases in abundance at the genus level. For example, Akkermansia, that is associated with intestinal health, and Turicibacter and Dialister, that are anti-inflammatory microorganisms. Also, we saw the relationship between microorganisms are different depending on the type of the donor and substrate being studied. As a conclusion, the structural variety of pectin and apple pomace influence the fermentation potential and the ability to modulate the intestinal microbiota. These results open the way to achieve its valorization through the formulation of new probiotics for particular population groups, and the use of both pectin and apple pomace favor the representativeness of some microbial population that decrease in IBD. Finally, I want to thank everyone who has made this work possible.
My name is Coral Barcenilla. I'm part of the University Lyon partner, and we've been involved in one of the work packages of the master project related to the design and validation of biopreservation strategies for meat products based on the use of lactidase bacteria. Foodborne outbreaks caused by the consumption of contaminated meat and meat products still occur with important negative effects on the public health. Listeria and Salmonella might sound familiar, as they are two groups of bacteria that can originate zoonosis associated with the consumption of meat products. For example, Listeria monocytogenes is a cause of major concern, as it has the highest fatality rate in Europe among the rest of bacteria. This, together with the increasing rejection of consumers against chemical additives or the demand of green label alternatives, has promoted the development of new strategies for the preservation of meat products. The use of beneficial bacteria, such as lactidase bacteria, or the products that they can produce, can be used as natural antimicrobials to compete with harmful or spoilage microorganisms, and so extend the shelf life of meat products. Our research group has studied the viability of applying lactidase bacteria in different meat products to control the growth of those pathogens. We've tried different products, different conditions, and different types of applications. For example, we have applied the bacteria on the surface of a sliced food ham, or mixed with the ingredients to elaborate a fermented sausage, or in marinades where pieces of pork were dipped in. To evaluate the effects of this application of lactidase bacteria on the products, we have evaluated different parameters such as the bacteria present, the color and texture parameters, if any important acidification occurred in the products, and also a sensory panel evaluated the final product. So the results that we obtained are shown here, some of them. We can see the growth of Listeria and Salmonella during the ripening period of a fermented sausage, and we can see that on the last day of the ripening, while there's still high counts of both pathogens, as we can see with the darker lines, when the lactidase bacteria were applied in the product, we can see that those pathogens were reduced, as we can see with the light lines. In addition, other bacteria that can cause the spoilage of food products can be controlled with the use of these lactidase bacteria. Procotrysterm specta, for example, is a well-known spoilage microorganism that grew spontaneously on two experiments performed in a fermented sausage and in cooked ham. But when the lactidase bacteria were applied into these products, there was a reduction of these spoilage microorganisms. So, as a conclusion, we can highlight that the tested lactidase bacteria demonstrated promising results to fight against different foodborne pathogens, that they were effective in different meat products and with various packaging conditions, and that the bacteria applied did not have any detrimental negative effect on the meat product quality. If you have any questions, we'll be happy to answer them.
Good afternoon, everyone. I'm Nicola Carlino, PhD student from the Segata Lab of the University of Trento, and today I will present you our project entitled Created Food Metagenomic Data, which is a large database of food microbial communities. But why do we need such a database? So we have known since many years that microbes have an important role in food, which can be positive, such in case of fermentation, or negative in case of sporage or pathogens. And up to a few years ago, it has been studied through classical microbiology methods, such as isolation, which had strong limits, for example, the difficulty or impossibility to cultivate microbes, and also the limitation to study one microbe at a time. Almost a decade ago, we had that technical revolution, which now allows us to study the metagenomes, which are the disordered DNA information of the whole microbial community. Meaning we have a lot of data, but in order to take out sense of it, we have to use a computational pipeline and a reference database. And that's what we did. So we built the first database of food metagenomes, CFMD, which is large and comprehensive, including over 2,500 samples from all over the world, almost 2,000 of which coming from the master partners. We have samples from more than 50 countries, 5 continents, and from very various food sources. And the master contribution was fundamental not only in terms of quantity of samples, but also in the increase of quality, that is to say, the information per samples, and also from food types, since we are now able to study foods that weren't available before. And that's our result. We have been able to reconstruct almost 300 new putative novel species, over a total of 3,600 species different found in food, among which we have been able to characterize them among those which were commonly shared and those that were type specific. But all in all, the bottom line is that communities are very food characteristic. So our result is the construction of a public database with the unprecedented contribution of master, whose main results is the demonstration that food microbial communities are very diverse and still in expansion, since we are discovering new species every day working on it. And these have very important impacts, both in the scientific community, as this can be used as reference, but also in the industry, since it's exploitable for food safety, quality control, and also the production optimization. But last but not least, you can also use to identify probiotic species and prebiotics. So thanks for your attention, and thanks to all our collaborators.
So I hope you enjoyed that. So 7 brief presentations to give you some of the research highlights of very many that were generated over the course of master. So we now have around about 10 minutes to go through a series of different questions and answers. I'll ask the various different panel members to keep their answers relatively short, because we do have quite a few questions. And what I might do is go through the questions in the order in which the presentations appeared. So I'm going to start off with you, Fiona, if you don't mind. So we have a question relating to, very simply, how were the bioinoculants applied?
Yeah, the bacterial bioinoculants were applied directly onto the seeds. So we just soaked the seeds, dried them, then they were distributed on the surface and then rolled in according to normal agricultural practice. For the mycorrhiza, what we did, we got it in a fine powder and we mixed it with sand and then distributed it across the surface. And again, it was rolled in. So that was what was available at that stage, but our partners had still since developed a further formulation of the AMF, so it may be that it's more improved now than what we used, but we used it as powder. Thank you for the question.
Super. And maybe another quick one for you. So was any consideration given to the suppressive activity of soil, or will that be important going forward?
Yeah, very, very interesting question. I'm not sure whether it's referring to the suppressive effect on potential pests and pathogens or the introduction of a bioinoculant, but I suppose the same concepts apply. Generally, it can be quite difficult to get if you're putting something into a very diverse microbial community, it can be quite difficult to get it to establish, whereas less diverse communities tend to be I suppose, more easily established. To answer the question, I suppose that we didn't specifically test this because the strains we have used hadn't been tested on grasses before. We're kind of confined to a few different soil types, but it's definitely a really interesting question. So thanks for that.
Super. Thanks, Fiona. I'll move on to Mattis. And for the question and answer session, we have Viggo Martinsen representing Mattis rather than Rene. So Viggo, Rene nicely presented the sustainability value of the intervention that you employed, but can you maybe comment about what commercial savings can be expected if waste streams are used as an alternative protein source for fish feed?
Thank you, Paul.
Hi, Viggo, it's hard to hear you.
Okay, do you hear me now?
A little better, yeah.
Okay. It's mainly 2 points that we are looking at. It's both from an environmental or sustainable point of view, and also from the cost-saving point. So both are very important, and maybe the sustainable point of view is more important than anything else. So using alternative proteins than fish meal in there for the feed is very important, which we are using side westering. Sorry, I have a little bit trouble of talking, but did you understand what I was saying?
Perfectly, super. Thanks for that, Viggo. Okay, so I have a question for you, David. So David, what was the quantity of the H2 acceptor that was needed to have an effect?
Sorry, can you repeat the question?
Yeah, it was what was the quantity of the H2 acceptor that was needed in order to have an effect?
Well, we're talking about reductions on methane that are around 20+% of the reductions, with increases in the hydrogen of around 20, 70%, even 100% of increase in the hydrogen concentration in the rumen to really have a real excess of hydrogen that can really make a difference in terms of the fermentation profile.
Can you maybe comment in terms of where it all is in terms of the TRL, or how long might it be before the product is available to the market?
Yeah, here we're talking about a combination of products and the inhibitors. So the first product of the combination, there are already products in the market like Bovar 3-NOP, which was recently approved by EFSA. And I expect that others such as Asparagopsis and other products will be ready in the market in the future. But the combination, it depends on what second component comes into the equation. So if it is a natural product that has a high concentration of these organic compounds, it could be a matter of 1 or 2 years because they could be considered feed ingredients. But if we're talking about concentrated materials or chemically synthesized materials that need to be combined with methane inhibitors, then I expect the process to take longer because it has to go through a salt technical additive process, which normally takes years.
I see, makes sense. Thanks, David. I might move on to Vincenzo. So Vincenzo, can you maybe comment about the SOP or the standard operating procedures that were used to map the microbiomes of the food industry? So those were obviously performed in research laboratories. Do you think that individual companies can do that, or will they need to outsource it, or where do you think the future lies?
Sorry, can you repeat? I didn't hear you very well.
So you described an SOP or an approach that was used in a research project in Master. So, but do you think that the swabbing-based approach with DNA sequencing and microbiome analysis, is that practical within a food facility? Would they need to have their own lab, or would it be easier to outsource that with a third-party provider?
Well, of course, it depends on the size of the facility. So I'm for sure for very little facilities, it's quite difficult to set up a laboratory and also perform DNA extraction and so on. But for larger companies, it can be quite affordable also in terms of cost of sequencing, which is continuously decreasing. And also it may be very easy to perform in a very simple lab. So very minimum equipment is required to perform swabbing and DNA extraction and so on. The problem, so the bottleneck of the whole story would be the data analysis, because of course there are some skills are needed to analyze and interpret the data that you obtain from the sequencing.
Yeah, that makes sense. Okay, super. Thanks, Vincenzo.
Thank you.
So Inez, I'm going to move to you next. So you described the development of a prebiotic ingredient from apple waste. So can you tell me about what's the best method for pectin extraction?
There is no better method than another. It depends on the structural characteristic that you want to obtain.
Okay. what do you think are the next steps to use pectin as a prebiotic and to, I guess, move towards the commercialization of this research?
Yeah, we are now carrying out the studies in mice, and later we will have to carry out more analysis to verify that it can be used as a prebiotic.
Okay, all right. Thanks, Inez. In the interest of time, I might move on to Coral. So, Coral, there's a few questions for you. I'll start off with one anyway. And so, there's a question as to whether these bacteria could be applied in products other than meat or meat products.
Yes, so the easy answer would be yes, but of course it needs more research. We will need to apply those bacteria into different products to test them, how they develop in those products. But I know there's more products rather apart from meat products that they can also be applied using biopreservation strategies. So yeah.
Okay. And another question was just how long can you extend the shelf life when you use the LAB as a biopreservative?
So I can say that the studies that we've done, for example, in the fermented sausage, we did the study for 36 days. And in the case of a cooked ham, the study was performed during 29 days. So that would be the period in which we studied this product.
Okay, great. Thanks for that, Coral. And then finally for Nicolo, there's quite a lot of questions relating to your presentation, Nicolo, and I guess you won't be able to we won't get through them all now, but maybe some of them can be answered in the question and answer panel in the chat function in a moment.
Sure.
But one of them relates to whether is this finished, or will the results be published, or what's going to happen with them now that you have all of this nice curated food metagenomic database put together?
Well, the database will for sure be public, and it will be made public as soon as the publication is ready. So it will be accessible by the whole community, and also hopefully will also be like having contribution for the community from the community.
Okay, and is that it? Is that the end of the database, or will it continue to live and be further expanded?
No, that's for sure the first release. The idea is to continue to keep it updated as soon as the new studies about food metagenomes will come out. So part of the work was also already a gathering of public studies, and this will continue with the life of the database.
Great. Thanks, Nicolo, and indeed thanks to all of the presenters and for making themselves available to answer those questions. So I will ask all of you to have a look in the Q&A function, and any questions that weren't addressed already to maybe type in your answers during the next session. So on that note, let's move on to that second session, and that relates to the various different innovations that came from Master. Again, just some examples, and I'll hand you over to the chair of that session, who is Lisa Mahler de Sanchez.
Thanks, Paul, and hello, everyone. So I'm Lisa Mahler, scientist in the microbiome product development team of QIAGEN, and naturally, we were very happy to be involved in this microbiome-based project. We contributed with a DNA extraction protocol aimed at expected low biomass samples for the various food industries. And we were also looking into the issue of host contamination derived from plants when investigating plant-associated microbiomes. Like Paul said, today I'm sharing the session of innovations. And regarding the innovations, I want you to know that the master project, during the project's runtime, was taking part in the European Commission's Innovation Radar, which is hosting a platform where you can find a list of cutting-edge innovations that are EU funded and were also developed or are still under development within the EU. And the master project submitted 21 innovations themselves, and all of them are now represented on the Innovation Radar. which is great. And in the next talks, we are going to see some examples of these. And for one of them, from the master beneficiary INOC, I want you to know that the innovation circling around the mycorrhiza products was selected by the Innovation Radar to pitch on deal flow, which is very nice because it's a European portal that connects EU-funded innovations with investors and corporates. That is, yeah, a real something very nice. All right, so we are looking forward to the talks that we are seeing now.
Hello, everybody. Welcome on my talk. My name is Eva Lusig from INOC. We are based in Germany, and we help to the transition to a more sustainable agriculture by developing biofertilizers based on arbuscular mycorrhizal fungi, even if many challenges remain for the routine use of these products in agriculture. So as a result of geopolitical and climate crisis, the conditions for food production have become more complex during the last years. We see an increase in the price of fertilizers, which are also less available, and record heat and drought episodes regularly affect plant growth and yields, at least here in Germany. And to make the situation even worse, the conventional agrochemical-based agriculture damaged the soil ecosystem functioning in many fields, like practices as application of high doses of fertilizers, fungicides, and pesticides, or intensive tillage, were all detrimental to plant beneficial microbe diversity and activity. So we really need now to restore our soils and go towards alternative cultivation practices. And the regulations in Europe also restrict more and more the use of phytosanitary products. And plant growth promoting microorganisms may represent one of the solutions. Arbuscular mycorrhizal fungi form symbiosis with more than 70% of terrestrial plants and constitute a key functional group of soil biota. And they can not only improve plant growth through increased uptake of nutrients, but have also non-nutritional effects by alleviating biotic or abiotic stresses in plants and in stabilizing soil aggregates and microbial communities. During the last years at INOC, we have developed a successful commercial product called Advantage, which is a concentrated inoculum based on colonized root fragments. But this product had absolutely to be optimized for its use in agriculture because the low volumes to apply and the powder-based composition represented huge bottlenecks for field application. And within the master project, we were able to develop clay-based pellets, which fulfilled the most important criteria of a formulation, which are to protect the fungal propagules from the soil influence, to concentrate locally the propagules, help to control the dosage of the product while being easily applicable with existing machinery, and of course, safe for the environment and presenting no inhibitory effect on fungal or plant growth. And after field validation on sunflower and corn, where really good results were obtained, as well on yield and drought stress alleviation, the product is now available on the market, and we hope in the future to test and adapt it as widely as possible to different crops and soils. Thank you.
My name is René Groben. I'm a project manager in the microbiology group at Martis, and I will give you a short presentation of the microbiome in aquaculture innovation. With a growing global population and the need for healthy, nutritious food, seafood is in demand as never before. However, current fishing practices put a lot of pressure on marine ecosystem, and the fish stocks worldwide are often in danger of overfishing, making them unsustainable in the long term. Aquaculture, which is the rearing of fish or other aquatic organisms in large tanks on land or in sea pens in the ocean or in lakes, has the potential to solve the problem and produce high-quality food in large amounts. As a result, aquaculture became quickly the fastest growing industry in the food sector. There are, though, problems associated with growing aquatic organisms in aquaculture, and one of them is the potential occurrence of pathogens among the high-density population of animals. A routine monitoring system for aquaculture pathogens is therefore necessary that gives the fish farmers fast and reliable information about if, which, and how many pathogens occur, and if measures need to be taken to protect the health of the fish stock and, of course, of the consumers. Current methodologies, like, for example, cultivation methods or immunological diagnostics, are normally focusing on only specific pathogens. They often take days or weeks and normally require specialized laboratories and highly trained staff. The Oxford Nanopore sequencing system that was tested as a monitoring tool in the master project has the potential to overcome these obstacles. In master, we developed a rapid protocol for pathogen detection and microbiome screening that allows for the early detection and prevention of diseases in the aquaculture industry. Through trials, adjustments, and improvements of the sample preparation method, which is a crucial part of the detection system, we were able to obtain high-quality DNA that could be successfully sequenced by the nanopore system. The advantages of the new monitoring systems are manifold. It is capable of identifying all microorganisms together, so it's not only focusing on one or two potential pathogens at a time. It works with a wide range of aquaculture-related sample types, like water samples, fish tissues, or, for example, the biofilters from the tanks. It does not need sophisticated laboratories and can be done on-site at the aquaculture facilities, so that there is no need to transport them to dedicated laboratories and waste time. The method is easy to use, and staff can quickly learn it. And last but not least, the whole protocol will only take a few hours until the first data is available, compared to days or weeks with traditional methods. This innovation in this new protocol was developed within the master project by researchers from Martis, Nanopore, and Livsglädje, and we would like to thank all partners for the fruitful collaboration.
Good afternoon, everybody. My name is Stuart Kerwin, and I was part of Work Package 3. And today I'm going to talk about breeding lower methane emitting cattle. So the objective within Master was to investigate the role of diet, host genetics, feed efficiency, and rumen microbiome on the environmental output in beef cattle. And one of those tasks was to develop an estimated breeding value for methane. So we know that methane is heritable and that we can select for it. And it's permanent, it's cumulative. So whatever we select for now, it has implications for future generations of livestock. Also, in relation to selecting methane, it's suited to situations in livestock systems where it's not suited for them, or basically that they can't implement feeding of dietary supplements that mitigate against methane. So we know that methane is linked to dry matter intake, so we need to be careful on how we select for methane, and that whatever we select for, that it doesn't have any negative effect on performance. So over the course of 2 to 3 years, we collected a large, I suppose, a large data set on animals. There was over 1,500 I suppose methane data collected on over 1,500 animals, and then there was also performance data that was linked to over 3,000 in relation to feed intakes and their performance, and that was combined to give us the first, I suppose, the first genetic proofs for methane. So essentially, what that means is that we can now actively select sires for reduced methane output. Just to acknowledge some of the people that worked on this work package: Sinead Waters, David Kenny, and Paul Smith from Teagasc. But also I'd like to acknowledge Stephen Conroy from ICBF who facilitated a lot of this work too.
Good afternoon to everyone. My name is Abelino Álvarez Ordóñez from University of León in Spain. And today I am going to talk to you about a very relevant innovation of our project Master, a protocol that we have validated for the mapping of microbiomes in food industries. The characterization of microbial communities that colonize food industries is extremely important, as these communities can have an enormous impact on the quality and safety of the food that we consume. Monitoring and surveillance of microbial contaminants has been traditionally approached through the cultivation in laboratory media and the detection and enumeration in culture plates. However, these culture-dependent approaches are time-consuming and only allow to analyze one microorganism at a time. In recent years, the huge developments in DNA sequencing technologies have granted the possibility of approaching the characterization of microbial communities without the need for cultivation, which offers many advantages as information on multiple microorganisms can be simultaneously obtained. However, food industry samples are challenging as they are expected to contain low microbial concentrations and may have residues of cleaning agents and organic materials. In addition, there are no protocols available in the market for the isolation of DNA of high quality from this type of samples. In Master, we have developed a complete protocol with this aim, which with standard operation procedures for all the steps of the microbiome analysis, including the sampling in the facilities, the sample manipulation in the lab, the extraction of the DNA sequencing, and the final computational analysis. Remarkably, our project partner Teagasc, a leader in the market of DNA extraction kits, has collaborated with the development of a new DNA extraction protocol adapted to the special needs of our food industry samples. The new protocol was successfully validated in more than 2,000 samples from food processing environments, from raw materials and products, and even from operators taken in 114 food processing industries of different production sectors from 5 different European countries. The development of this innovative protocol, which we hope will have a very significant impact for food industry operators and also for analytic laboratories, has been possible thanks to the collaboration of different partners of the Master project, including Teagasc, the University of Naples in Italy, Teagasc in Ireland, Foxy in Austria, CSIC in Spain, Matis in Iceland, the University of Trento in Italy, and our University of León in Spain. To finalize, I would like to thank all the food business operators that allowed us to test our protocol in their facilities, and I would also like to thank you for your attention. Thank you very much, and good afternoon.
Good afternoon, everybody. My name is Laura Nine, and I'm a postdoctoral researcher in University College Cork. And I want to give you a brief overview of the work that we have carried out during the Master project. And our task was the upcycling of brewing byproducts to produce functional food ingredients. So we were working mainly with 2 brewing byproducts. So the first is barley rootlets, which are obtained from the barley malting process. And the second is brewer spent grain. Brewer spent grain is the most abundant byproduct of the brewing process, with about 36 million tons of this produced globally each year. Currently, these byproducts are used as animal feed and sometimes disposed of in landfills, which is quite unsustainable. However, they represent a really valuable source of nutrition, containing about 30 to 45% dietary fiber and between 30 and 40% protein. So to date, the addition of these byproducts into food as a nutrition source has been challenging due to the negative effects that they can have on food quality attributes, such as flavor, texture, appearance, and overall acceptance. However, one method which can improve the performance of these types of byproducts in foods is fermentation. So in our research, we have seen that the use of fermented ingredients in foods, they have a better performance in food, they can enhance the nutritional aspect of a food, they can improve the sensory attributes of a food, they can extend shelf life, and they can also provide health benefits to the consumer. So in our task during the Master project, we used fermentation technology to upcycle brewing waste materials into functional food ingredients. So we developed a range of different fermentation protocols, and we tested various different lactic acid bacteria, each which had their own unique characteristics. And with this, we were able to produce a range of functional fermented ingredients, which can be applied in a variety of different food matrices. So we looked at it firstly in an application in bread, and what we saw was that interestingly, each ingredient had a different function in the bread. So they all improved the fiber content of the bread. Some improved the volume of the bread and gave a softer bread with less hardness. Others increased the shelf life of the bread, and some gave a really nice sourdough bread taste profile. So what we concluded is that we can use fermentation to produce innovative upcycled food ingredients which have a better performance in foods and can be applied in a range of different foods. And what was really interesting was that we can tailor the fermentation in order to produce ingredients which all have different functions depending on what kind of food and beverage you want to apply it in at a later stage. So thank you very much for listening to this short presentation. I would like to thank everyone in UCC, which has worked on this project for the last 4.5 years, and also our collaborators in AB and Bev for providing the raw materials and also for their scientific input during the project. And I'll be happy to answer any questions you have. Thank you very much.
Hello, everyone. My name is Fernanda Godoy Santos. I'm an animal scientist and a doctor in agriculture microbiology. Currently, I work at Queen's University Belfast, and today I'm going to provide you with a brief overview of our work in the master project, where our group of researchers are creating new bacterial communities. So, as you have seen in previous presentations, microorganisms play a crucial role in agriculture, human health, and in the environment. To better understand bacterial dynamics, interactions, and functions, scientists have created mock communities. These artificial populations mimic the natural population, serving as simplified models for studying bacterial behavior in a controlled environment. In our bodies, microorganisms are everywhere, and their population diversity is influenced by the environmental conditions. Here, we have two examples, a median plate showing aerobic microorganisms from the skin, and another median plate showing anaerobic microorganisms from the intestine. Looking at these pictures, you can clearly observe the difference between the two populations. In addition to their role in understanding bacterial dynamics, interactions, and functions, mock communities have several potential applications. First, they are important in ensure representativity. As we mentioned earlier, microbial population can vary depending on the environmental conditions. Therefore, scientists carefully select specific bacteria to create mock communities that represent different types found in the natural settings. Mock community also play a crucial role in quality control and innovation. They allow researchers to predict and assess bioprocess performances, and also offer exciting possibilities in food product development and innovation. By manipulating the composition of the mock community, scientists can optimize the characteristics of the final product. This can lead to the creation of new flavors, textures, and even improve nutritional profiles in food. Another significant benefit of the mock community is their contribution to the research reproducibility. Scientists can compare and evaluate different techniques using mock communities, ensuring reliable and consistent results that can be replicated across experiments and studies. In this context, the scientists involved in the master project focus on creating two bacterial mock communities, one aerobic mock community food-associated, and one anaerobic mock community rumen-associated. Throughout the project development, new bacterial cultures were isolated, characterized, and carefully selected to construct these two mock communities. The above mock communities were analyzed using genomic analysis, and the preliminary results confirmed their quality. So here, I just want to say thank you for your attention, and thank you to all our collaborators.
So congratulations to all the researchers for their very impressive outcomes. I'm going to try to follow the good example of Paul and go in the order of the talks that we have just seen with the questions. If I'm not managing this promise, then well, excuse me for that. But we are starting with Eva. So the mycorrhiza-based products, how compatible are they with conventional phytosanitary products?
Yeah, so thank you for your question, Lisa. It is indeed an interesting and complex topic, and we are currently involved in another EU project to study this aspect. So what I can say is it's pretty clear that high doses are detrimental to mycorrhizal diversity and the functioning of the symbiosis. But even if it seems surprising, there is in fact no real consensus on the impact of phytosanitary products in cropping systems when they are used at the recommended field rate. And we can observe various effects ranging from negative to neutral and even positive. And to complexify the situation, these results are often contradictory for one given product between different crops and fields. And it is important to note that both chemical and plant-based products can affect the symbiosis. So for example, we have neem extracts used in organic farming that can affect the colonization and the communities as severely as a chemical counterpart. So in fact, there is no clear separation between what would be a good organic compound and a bad conventional chemical product. The effects are really substrate, dose, plant dependent. So as take-home message, I would like to say that AMF-friendly soil management is not incompatible with conventional practices, and mycorrhiza should definitely not be considered as something which is limited to organic farming.
I see. Thank you. Okay, we're moving on to Vigo for René. There was a question if this method that you have developed can be also applied to other purposes than in aquaculture.
Thank you, Lisa. Yes, it can be applied to for different application than aquaculture, but although the nanogrowth technology is very well established, we always come again to how to process the DNA extraction and preparation. So again, we need high quality of DNA, and that can be a tricky part. Depends on what you are analyzing.
Do you actually think that the high molecular weight extraction is making it
better? Yes.
Yeah. Okay.
Yeah. So it's very important to get that.
Yeah. Yeah. I understand. Okay. Thank you very much.
Thank you.
Okay. Then we are moving on to Stuart. Is there or was there in the end a breed that was clearly better than the others?
Hi, Lisa. Stuart actually has had to step out. It's Sinead here. So yeah, for our study, like we looked across 1,600 beef cattle. Some of them were crossbred. And just in this study, we could see that the limousine cattle were coming out as having lower methane emissions marginally. So this would have to, I suppose, validate this in larger populations going forward. And also these genetic evaluations would also have to be validated outdoors in pasture, so basically in grazing systems. But this is really a first excellent study to show we can breed for lower methane emissions. And it was done in a very controlled indoor setting. And now we're going to bring that to, Ireland is largely pasture-based country. So we're going to bring that to an outdoor setting. But in terms of the breeds, yeah, limousine is looking to be a lower methane emitter.
Okay, great. Thank you. Mm-hmm. What do you think how long it might take to bring that breed in the end to the market?
So right now, the Irish Cattle Breeding Federation have announced these, I suppose, proofs are, so these will have to be, as I said, validated. And that will start coming into effect over the next few years. So maybe, 5-10 years, we should see these being, I suppose, used throughout the country. But first of all, we'll have to validate them ourselves in Teagasc and working with our partners, research institutes, and also then been implemented slowly onto farms, commercial farms and farms across the country. But I'd imagine we'd make very good progress in 5 to 10 years.
Great. Good luck.
Thank you.
Okay, so we are moving on to Avelino. There was a question, how if the protocol that you have developed is already accessible?
Yeah, thank you, Lisa. So the protocol is being at the moment considered for publication. We're in the process of revision. So hopefully it will be available publicly in a couple of months, hopefully. And of course it will be including all the steps from sampling to all the manipulation in the lab, the DNA extraction with advances made by you, by Qiagen, and with all the sequencing and computational analysis. It will be the full pipeline from sampling to bioinformatics. So yeah, hopefully by the end of the summer, this will be available for everybody.
Great, very impressive. I will also comment on a question that was actually probably directed in my direction. The protocol that we developed here in Qiagen is not yet a kit, but we are planning to turn it into a kit, but it's going to take more time. If you're interested in getting this customized solution at the moment, please contact me. All right, then Laura. There has been actually a very long question. I have to read it out. I'm sorry. Did you use known cultures or bioengineered cultures of microorganisms in your upcycled byproducts?
We didn't use any bioengineered cultures. The cultures that we used were from our old culture collection here in UCC. And we picked these specific ones based on their characteristics that we already knew from working with them before. So some were antifungal producers, some were exopolysaccharide producers, and so on. So they were selected on the basis of their characteristics to see if those characteristics could also be imparted into the ingredient and then into the food matrix in the end.
I see. And have you combined that with commercial enzymes?
In the fermentation of the brewer's spent grain, we did. That was a combined enzyme hydrolysis and fermentation process. Yeah. So yes, we did. Yeah.
Okay, great. And how high can you go percentage-wise? So how many of the upcycled byproducts can you add in the end to food?
So what we showed there in the bread matrix, that was using the rootlet ingredients. We saw that about 5% was the optimum. You can go higher than that, but then you will have to play around with your processing and stuff to optimize that. With the BSG, we did do it at higher levels. We added it into levels in bread to reach a source of fiber and a high fiber claim. And I think the high fiber claim, it was added in levels up to about 15%. Yeah, but again, you will have to optimize your processing conditions there to optimize your product in the end.
Sure, makes sense. But I mean, 15%, it's already quite high.
Yeah, it is.
Great, thank you. Okay, and then we're going for the mock communities. And as far as I know, Federica is here for answering that, right? Okay.
Yes, I am.
Okay, and there has been, I think, also a question in the chat. We start off with if the mock communities can be used in food industries. So do they contain dangerous bugs or?
Yeah, this was one of the requirement of the mock community, since it's based on food chain associated bugs. So we tried to select isolates that didn't contain any antimicrobial resistance genes or virulence genes. So it was obviously we sequenced all the strains, and this was tested by our bioinformatician. So of course, ideally in the future, it could be something used also in food industry without any harm. Yeah.
Okay, and then can you comment on how you maybe more in detail used your mock communities?
So the main point was that when Master started, there was not really a mock community specific for food chain environments. So the idea was to generate and create this mock community with strains that, of course, are relevant for the food industries and food production. So, of course, the main aim would be to standardize experiment and testing when this occurs in different, for example, countries or in different laboratories. And this was an example in Master where we had the same, for example, the same test, the same experiment in different performed by different partners in different laboratories. This was so it's essential to have a mock community as a standard, as a control.
Great. All right, thank you very much. And with this, we are at the end of this question and answer session. And I'm handing over to Tanya for the next session.
Yes, thank you, Lisa, and warm welcome to everyone also from my side. I will now be using like approximately last half an hour of this webinar to guide you through the panel discussion entitled Beyond Master, the Future of Microbiome-Related Technologies and Their Applications in the Food Chain. So I would like to ask all of the panel members to put on their cameras, and I will briefly introduce them to you. So we have with us Dr. Angela Sesic, who is head of Competence Unit Bioresources at the Austrian Institute of Technology and has pioneered plant-associated microbiomes and is interested in understanding the interactions between plants' microbiomes and the environment and developing applications in this field. Then we already had him today, the Professor Avelino Alvarez Ordonez, who is principal investigator at the Department of Food Hygiene and Technology at the Institute of Food Science and Technology at the University of León and a member of biohazard panel of EFSA. Then we also have a Professor Christopher Creevy from Queen University of Belfast, who is a professor of computational biology. And his research group develops de novo computational strategies for metagenomic and metatranscriptomic data for the natural microbial communities and their hosts. Then we also have some members of our advisory board. So we have Dr. Colette Short, who is an independent regulatory specialist and registered public health nutritionist with over 30 years of industry experience gained in European and global scientific and regulatory leadership roles. We also have with us Dr. Gerald Coyne, who is an associate director for applied industrial markets at Oxford Nanopore Technology. We also have with us Mrs. Kelly Eversole, who has a long-standing experience in building private partnerships in agriculture, genomics, and agriculture research. And she's also established a consulting company, Kelly Eversole Associates, which focuses on enabling agriculture, science, and technology. And she's also serving as an executive director and board chair of the International Alliance for Phytobiomes Research and the International Wheat Genome Sequencing Consortium. Then we also have with us Dr. Murian Egan from the Teagasc, who is a communication officer for Teagasc Food Research Program, responsible for communicating and dissemination to various target audiences, including scientists, food industry, other public agencies, research organizations, journalists, and other stakeholders. And last but not least, we also have the Professor Paul Cotter, who was a master coordinator and is head of the Department of Food Biosciences at Teagasc, a molecular microbiologist with research focusing mainly on the microbiology of food, food processing and production environments and the gastrointestinal tract. So you see, we have quite a broad expertise here on panel, and I also warmly invite the audience to ask them the question. You have no unique opportunity. So please add your questions in the Q&A. And just to sort of warm up a little bit, building on this brilliant presentations that we have heard in last hour, all of the research highlights and innovation highlights, I would like to ask people on the board from industry, so which of these outputs that we have heard stands out for you, and how do you see their impact in the next 5 years? So perhaps Colette, if I might challenge you to kick off the round.
Okay, thank you, Tanya. I think first of all, I just want to congratulate all of the researchers for 20 innovations in the radar. Well done. I think you should really clap yourself on the back. That's phenomenal. I'm actually going to say what stands out to me from the master's project is the abundance of soft and transferable skills that have been gained by the researchers during the master's project, in addition to the technical skills. I've seen developments in communication, presentation skills, critical thinking, planning, and the ability to collaborate. And these are phenomenal skills that are often overlooked as outputs in projects. And I think they're a testament to Paul Morid and the leadership team, who really developed a very dynamic and supportive and inclusive scientific culture during this project. And I think these soft skills will pay dividends in the future for all the researchers.
Thank you, Colette. This is a really interesting aspect. So Kelly, would you also like to comment what stood up for you in the...
Yes, I'm happy to. And thank you very much for this opportunity to participate today and to actually watch this fabulous project go from start to finish. It's just been amazing. And I don't think it's the finish. So I think it's actually the beginning of this next phase. To me, one of the things that stands out the most is the breadth of the project, really taking it from that concept of One Health and really going all the way through the entire food chain. I think that is very significant. And it also proves that anyone who said it wasn't possible, they were wrong, because it is possible to do that. And I think that is really going to help set the stage for really embracing all this complexity and pulling all this together. I think the other aspect that is, to me, was really important was the transdisciplinarity approaches to the various problems. That's really hard to achieve, and particularly in these environments that are very complex. But I thought that the teams have done a phenomenal job, and I wish that we could replicate that all over the world, just globally, and have it being done in different efforts. So maybe that's the next phase for that. Thank you.
Thank you, Kelly. Gerald, would you also like to chime on this?
Yeah, absolutely. Thank you, Tanya. And first off, absolute pleasure to be a participant in the master program and to establish some great new relationships through the program. I suppose as a representative of Oxford Nanopore, we do have a particular perspective related to molecular analysis. And I guess a couple of things stood out. Firstly, I do think it's absolutely critical to have develop a curated database for microbiomes. I guess it's obvious that the effectiveness of any subsequent screening will only be enhanced through the generation of this database. Also, I, of course, was very impressed with the work from Mattis in Iceland, and this was obviously the development of a microbiome and pathogen screening tool that can be field deployable for the aquaculture industry. I think the ability to deploy close to source would be very, very important, because it really does increase the agility for the industry, and obviously into the future it could really improve fish production, and it can also have I suppose an economic impact. And as Viggo mentioned, of course, this could be deployed not just in aquaculture but for other sectors as well.
Thank you. So I would like to sort of chime in on this last reflection given by you and the food database, and perhaps a question back to the master team members. So how do you see this food microbiome database? How do you see it will impact the research, but also the industry in the next few years? And what do you see? What do you think are the next necessary step to really pave the way from implementation? So I don't know if Chris, you would like to start on that one, and then perhaps Paul or whoever would like to take it.
Sure. I think one of the most important things I think that's come out of this for the future is it gives us the references in order to understand how these microbial communities change in different situations. So what we have generated now is the baseline upon which we can compare other situations. So for instance, if there is a dysbiosis or a change in some way, we can utilize these in the future for identifying early warning signs and to take remedial action in those situations. So that's how I see where we can build upon these databases in the future.
Thank you, Paul.
Yeah, so I think the availability of the database has the potential to be really disruptive in terms of the next generation of where we will go in the future for microbiology testing with respect to safety and food quality. And that's really because in the food industry, there's probably still an over-reliance on old traditional agar-based approaches that have been used for decades, if not centuries. And we know as microbiologists that quite often those are not very selective. And In other instances, they're too selective in that you only find what you go looking for. So there might be a pathogen or a spoilage agent in your plant, but unless you know how to grow it or how to target it, you're not going to find it. And indeed, some of them are just not very easy to grow full stop. So I think this paves the way for the future application of microbiome-based tools so that using the protocol described by Avellino and the kits developed by QIAGEN and so on, that it's now practical to collect the samples and to do the sequencing, but the database is the key final component. If you don't have a good database, you can sequence forever, but you won't know what microbes that represents, and that's the key component. So I think it really can provide us with a new world, a new dawn, where microbiome technologies can be used extensively in food processing facilities and beyond.
Thank you, Paul. And I would just like now to use the opportunity, because we have an EFSA Biohouse member on board, and perhaps Avellino, would you like to comment on how do you see perspective, because Paul now also addressed the pathogen, so how do you see the perspective on introducing the microbiome in the food safety assessment in the future?
Yeah, thank you, Tanya. So I think there are different ways where how microbiome science can support risk assessments. No, one possibility would be considering possible harmful effects of different substances, additives, any particularity that is introduced into the food chain in particular microbiomes, let's say the human microbiome or the soil microbiome, the animal microbiome, etc. That could be a possibility that is started to be considered. The other way would be to use microbiome data to predict the fate, for instance, of particular hazards or particular microbes in the food chain, in different food chain scenarios, which could provide very relevant information for exposure assessment or for hazard characterization or risk characterization in risk assessments. No? In both cases, anyway, I think there is the need to try to develop new methodologies to try to exploit that kind of data in quantitative risk assessments, because risk assessments are mainly based so far in trying to model how a particular hazard behaves or how it changes or how and now within these new technologies we'll have to look at much more complex data sets and how to exploit that. But I know that, for instance, EFSA is doing some work in that regard. In the last years, it has been funding a few explorative projects trying to look at how the microbiome data can be introduced in risk assessments. And also there has been a quite recent scientific opinion on how these metagenomics and genomics technologies can be exploited for outbreak investigation or for resource tracking or for risk assessment. So actually, I'd say that more and more we will be hearing about how this can be incorporated in risk assessments, and hopefully in a few years it will be more common.
Thank you. So I see that definitely the food database and new sequencing technologies raise a lot of interest, but since master was so broad, I would like to encourage audience also to ask about the other fields. And now I'm going to move a little bit back from this and go to other innovations that we have. We heard a lot about the bioinoculants, about different fermenting cultures, etc. And I would like now to ask perhaps Angela, the microbiome technologies have been now in the spotlight in the last year. What do you see is now the biggest bottlenecks for the implementation and exploitation of these innovations in the future? So what is still in front of us?
Yeah, thank you, Tanya, for the question. I think one big bottleneck, but of course it's just one of several, is I think the issue of microbiome literacy. So if we want to have microbiome innovations and applications be used and accepted by consumers, by farmers, by the policy sector and so forth, I think we need to take care that there is more awareness of microbiomes. So a consumer has to understand roughly what a microbiome is and that it's very important. And also policymakers and regulators and farmers, they have to roughly understand what it is. So I think we need to improve along these lines. And this is also one of the many aspects we will address in our new microbiome support association, which is currently set up and which is somehow the continuation of the CSA microbiome support. I think other bottlenecks are, of course, the regulatory sector. So many of the current applications are already challenging to find suitable regulations, so to allow, a safe but still quick, market introduction. But also things like scaling up, I think, is one of the big bottlenecks, and we really need really successful and convincing applications to really spread the importance and the potential of microbiome applications.
Thank you so much. And yes, I think we all learned how communication can be challenging and how much it is being expected of us scientists in the last years. But we have a communication expert on board. So perhaps you would like to chime in, and perhaps could you share with us what do you think or what do you believe makes for effective science communication? How can we use it really to bridge the gap and to bring this awareness that Angela now sort of highlighted that's so important? So Muriel, if you would like to enlighten us a bit.
Thanks, Tanya. Following up on Angela there, I think one of the great things about mastering projects like this is the whole area of microbiome research does garner attention from the media, from the public, and from the industry. It is one that people pay attention to. So on the one hand, that's part of the challenge is getting attention, and I think we can do that. On the other hand, there can be a lot of noise, opinions, and it's up to the scientists that they can present and communicate their key findings in a succinct, concise manner, which is the challenge. In terms of bridging the gap to the stakeholders, it's about knowing the audience, really. You mentioned policymakers earlier. If you're speaking to them, you're kind of looking to have actionable recommendations that they can take on board. If it's farmers, the agri-food industry, it's about, what are the innovations and how soon can they implement them, and what are the tangible benefits? Whereas maybe then for the public, it's kind of more the story of the why you started this research and what have we learned and how will it affect their kind of lives and society, maybe in a broader sense. So it's about thinking about that audience, and then also it's about being able to communicate it. I said succinctly a number of times, because for most of these conversations that you've had, and I've seen it at events and with industry, you've a few minutes really to get your message across. So you need to know your key finding and what it means, the so what of it. If you have a bit longer, you can go into your methods and the kind of longer side, but you really need those 3 or 4 sentences to get the attention straight away.
Thank you. So I guess you approve of the work that was done in the first 2 sessions.
Very much so, yeah. It's a good example of how
I admire all of the people, because this is always really the challenging to bring your message in across. We have talked and we have heard at the beginning from Colette and from Kelly a lot about what they were inspired also by the transdisciplinarity and the width of this project. And we did actually really involve many different stakeholders. So I would just now like to pose an open question to the round, and then perhaps Paul as a coordinator can kick it off. So how do you think we can build on these lessons learned? How can we extend the transdisciplinarity of our research? And also, how do you think we can open, beyond Europe, so going a little bit more on the international platform? So Paul, if you would like to start, and then anyone who wants to chime in, please.
Yeah, so I think it's important to do so. I don't have one answer, but I think it'll be a combination of different approaches. So the partnerships that were established within Master, because they're so broad, there isn't a Masters 2, so to speak, that will continue the research altogether. But there are a number of other projects within Europe funded by the EU and through other schemes that will involve very many of the master partners and other people that will allow this work to continue. Several in the rumen space, in the plant space, in fermented food area. So I think that will continue. I think the aforementioned microbiome support foundation that Angela referred to will be very important as well, and will be an opportunity for like-minded people to get together and discuss and to plan and make future plans as to how to continue research in this area. But I also think, though, that by taking the approach that we did, whereby we're making the results publicly available, we're sharing and disseminating, that we'll encourage others to want to join and to reach out and hear more about Master and to employ some of the innovations, some of which are already being developed with a view to commercialization, but others, there still is a potential for other industry partners or other academics to get involved. So I think there will be a multitude of different things, but in all sorts of different directions.
Thank you, Paul. Kelly, you are our international member of the panel, or non-European member of the panel, and you are always really involved in our, keeping a track on European project, involved with European researcher, even though you're located stateside. So how do you see this, managing it internationally, bridging the gap, bridging the researchers, making the research more coherent? Would you like to comment?
That's really, that is definitely kind of the vision of the International Alliance for Phytobiomes Research, is to really build off what's going on in different countries or different regions, like with Angela's Microbiome Support Association. And we're starting to really see some ideas gel. That one of the challenges that we try to do is not only look at the microbiome space, but then try to bring together the other components of a phytobiome. So we're really looking at, can we begin to really link seed genetics and get plant breeding companies engaged, and we're really trying to move in that direction. We're also trying to now look at controlled environment agriculture, which may build off of some of the stuff that you have created and some of the discoveries, and trying to put that at an international level. So we're kind of looking at ways to both capitalize on the microbiome with specific multinational and in some cases bilateral projects. We are seeing funding agencies that are more receptive than they have been in the past. So there are all kinds of different projects that are going on between countries or opportunities, and we're trying to use that as a way to build these international bases. And I think the other thing that we try to do is visibility. We've had several of the master program leaders on our webinar series, and we hope to try to do that more and cover other projects, other efforts underway. So I just think it's going to take all of us kind of working together, and hopefully under of course I'm biased, under the International Phytobiomes Alliance umbrella, and to have us be a way to kind of merge it together. I will say the One Health aspects and the bringing in the rumen projects with the forage system, that's fabulous and phenomenal. And I think that's really ready to take off at an international level. It's one of those areas that could really benefit tremendously. I think the challenge that we will all have, and it'll be a challenge even with the database, is the idea of, can we all get together and try to use the same protocols? Can we try to use the same database and put all of or at least make them to where they can be interchanged? So I think those kinds of things are hard to get. And, in my efforts on wheat, we never achieved one single database. We have our International Wheat Genome Sequencing Consortium database, but not everybody will use it. Everybody has their own database. So I think, and that's just with one crop. So you're dealing with, the complexity here is going to be a big challenge, but it's one that I think you can, the experience you've built here, the partnerships that you've created could help lead this effort and prove that, you don't just have to reinvent the wheel every time. We can try to work together. So I hope I've answered that. It's a very complex question. So.
Yes, thank you, Kelly. You really highlighted the complexity of the microbiome and everything which we still have ahead of us. And what I personally also liked about the master was the opportunity. We had a lot of PhD students and postdocs in the project, and we have now so many experienced people on the table. And I would just like to ask you, what one piece of advice each one of you would give to a young scientist that want to pursue the microbiome-related research, being it in agriculture or food, going into industry, staying in science, turning into science communicator, or perhaps going more into the, this regulatory area? What one piece of advice you would give them? So I will just start from the top of my screen and start with Paul, and then I hope that everyone can chime in with something.
I will give the same advice that I give to any PhD student, regardless of what they're working, and that is to work on something you feel passionate about. Because if you work on something that you're enthusiastic about and you're inspired by, then coming to work each day won't feel like work. It'll be a joy. And maybe that's a bit maybe a bit naive of me, or maybe it's a bit idealistic, but that's the approach I take, and it stood me in good stead. So that's the advice I give to others as well.
Thank you, Paul. Colette?
I would say make connections, build networks across disciplinary lines, build on the master's network, share information, because I think going forward, partnerships and the sharing of scientific information is where we're really going to make impacts.
Brilliant. Thank you. Avelino?
Yes, I would say maybe try to think a bit ahead of your very particular project and see what is going to be the impact of your research and the transfer possibilities to try to be in a more, let's say, expanded way, aware of what your project means and what are the possibilities for the future as well.
Thank you. Gerard?
Yeah, well, I say go for it. Microbiomes, it's a new frontier for food and ag, and as a new frontier, as we see, there's lots of innovation, and innovation is going to present opportunity. So I'd absolutely recommend it for any postgraduate moving into industry.
Good. I'm a little bit biased, but I fully agree with you. Thank you. Chris?
Yeah, I would just say that, when you're doing a project like this, to recognize that all the skills and all of the things that you're learning to do and developing are eminently transferable to other areas and other working spaces and other careers, and to take a step back and look at the set of skills that you're learning and developing over your PhD or your project, and make sure you recognize that when you move on to your next steps.
Thank you so much. Murienne, perhaps a little bit different.
Yeah, from my perspective, I'd be telling people to just, students to just get out and talk to people, whether it's at kind of take the chance to go to a science festival or, even like standard conferences, scientific conferences, speaking to people in a kind of natural way that's not a presentation. It will help your communication skills for those future presentations and vivas, but you also never know what you're going to get back out of it, whether it's from talking to another scientist or even, if it's the public. The level of interest that's out there is huge, and you never know what kind of little comment or thought or idea might be planted just through those conversations. So it can be scary at first when you're speaking about your work in this way, but it gets easier the more you do it, and it's very enjoyable once you get into it.
Thank you. That's a really valuable piece of advice. Kelly?
Well, yes to everything that everyone has said before me. I think those are all extremely important things to consider. There's two things I would add to that, and one is try to get some industry experience, because then it makes you more flexible and more marketable, whether you're going to stay in academia or whether you want to move in different aspects of the industry. And the second thing is soft skills. Get as many soft skills as you can get while you're doing your studies. take leadership courses, learn about what your purpose is, like as Paul was talking about, to get that passion, but tie that into overall leadership. So those are really the two things that I would recommend that everyone really try to do.
Thank you so much. Angela, I know a lot was said, but I hope you can still add a little bit on.
Yeah, I'm not sure I can add anything more. I mean, there were so many important things said. I think I would also, on the one hand, emphasize the importance that you need to be passionate about the complexity and diversity of microbiomes. I think that's a very fascinating world. But on top of this, as Kelly said, the development of soft skills, to really be able to communicate with others, to take responsibility, to act as a leader, and so forth. I think these are also additional components. So, but it's a combination of everything what has been said. So, love the diversity and the complexity of this microbiome world.
And don't give up. Yes, thank you.
Never give up.
Yeah. So it's a brilliant round, and I think I could chat with you probably for the next 3 hours, but our time is running out, and so I will wrap up with my final question, and we will just now turn it around. So Angela will start, and Paul will finish, so to make it a little bit more fair. And you know how they always say we are all visionaries, each and every scientist is a visionary of a sort, but I would like to ask you, what is your vision for the future of the microbiome related technologies and their applications in the food system? Like, what would you really like to see happening or happened in the next 10 years, and where, giving a little bit inspiration? I do hope that we have young scientists in the audience giving a little bit of inspiration where this, this journey could still go. So Angela, please, if you could start on that one.
Yeah. On the one hand, I think it would be great if we all, the farmers, all stakeholders are really aware on the importance of microbiomes, and include this thinking or this fact that microbiomes are everywhere and are doing something in their applications. So that, let's say, in the farming sector, for instance, it's not only the soil physical and chemical parameters, that it's also the biological parameters, for instance, and that we come up with more precision applications. So whether it's precision personalized nutrition or precision farming or precision medicine, I think microbiomes will play a very important role in all these applications.
Thank you. Kelly, you're now the next one.
Yes, so I definitely agree with Angela, and I wrote it down, and then she said it, so I won't repeat it. But I do think this vision of having the microbiome an integrated part of the future is critical. And to me, from an agricultural standpoint, if we want to address climate change, if we want to address methane emissions, if we want to look at carbon sequestration, we have got to integrate the biological aspects of all along the food chain to combine them with all the physical, the geophysical components. And only by doing that will we really begin to get to sustainability and to, maybe will we have in situ, in field, little strips tied to the tractor by 10 years? Probably not quite, but I'm hoping that we have infill sequencing much more rapidly proven. As you know, I'm very interested in the infill sequencing aspects. So I'm hoping that will help us get there. And so I think we'll be part of the way there in 10 years.
I definitely hope so. Thank you, Kelly. Muren.
Yeah, similar to the comments before, I just look forward to seeing and hearing about how the microbiome technologies and innovations we heard about today are going to contribute to those future food production and food sustainability problems. And, I look forward in particular, there was a couple of interesting talks about upscaling and, making high value products out of food waste and that. And that's an area I'm really looking forward to seeing how that plays out.
Thank you. So now going a little bit from agriculture. Oh, my image is shifted from agricultural, but back to perhaps more technical. Chris, what's your vision for the future?
I think I would love to see us move more and more towards, move away from considering microbes as individual actors in an environment, an associated environment, and going more towards this idea that they are an integral part of the microbiome and the system that they are found, the environment that they're found. I think that we've taken quite a few steps towards that in this project, and I think that there's quite a ways yet to go and a lot of unanswered questions in that space still.
Yes, definitely. But thank you for your input. Gerard.
Yeah, well, a short tagline for the vision. I see the democratization of microbiome data and microbiome related products. Of course, that data and those products is more accessible than it has ever been. I think it is important that that trend continues. An important aspect, I think it's already been mentioned, is around the making easier the data analysis and the interpretation. That will make it more accessible to the masses. And I think, as referenced, I have a keen interest in in-field sequencing, and I do think that data analysis piece is an important contributor to that.
Thank you so much. Avelino?
Yeah, so from my perspective, coming back to food safety, food processing, maybe other perspective to the ones that we have heard in the last minutes, I would say that I would expect to see more microbiome diagnostic technologies in the food processing, so applied by food industries, but also maybe to of course continue having programs and monitoring programs focused on controlling particular microbes and hazards that could be still very, very relevant. But I would think that maybe in the next few years, I would expect to see some movement to not only talking about controlling particular hazards, but about preserving microbes and preserving ecosystems and considering the presence of these hazards in an ecosystem perspective also in the food industries. So whether some strategies can be produced and developed to control the ecosystem and not only to target specifically a given microorganism. So I think that in food processing and in general and for food safety, in a food safety perspective, I think would be very, very relevant for the next few years.
Fully agree. Thank you so much. Colette?
So my vision would be that those involved in microbiome research wouldn't see regulations as hurdles, because I think at the moment a lot of people do, but I'm really positive about that because I think we're already seeing a lot of approvals. For example, in Spain, there was a vaginal product there recently, and we've had some pharma products in the US. So I think this is really important if we want to accelerate innovations to the market and really make a public health change.
Thank you so much. And again, we see how broad the microbiome-based technologies can be and how diverse impacts even the small group of people envisions. So once more, thank you all for this really, really nice discussion. I'm really sorry that the time is out. And I would also like to say thank you to Paul, not only for this, for this webinar and this opportunity, but also for the whole master project. And I will just now give to you to give your last piece of vision after Beyond Master and then to close event. And thank you very much to everyone.
Super. Thanks, Tanya. I guess given it's a wrap up as well, I'm going to answer the final question in a very broad way. And I'm going to use the metaphor of the smartphone industry, of how I guess when I grew up, there were no such thing as smartphones, and eventually, so I resisted them for a while, and eventually I started to use one, and now I'm addicted and can't live without it. And see children on planes, the infants who are now swiping and doing things on their smartphone. I'd like to think that microbiome-related technologies in 10 years' time will become routine and become part of everyday life, and the different types of applications will almost be the equivalent of different apps, just different ways in which we're applying the microbiome. And so that will include like DNA extraction and bioinformatic tools and different strains and consortia and bioinoculants and mock communities and media mitigation and waste valorization and all of the fantastic things that we heard about today. But so that's my aspiration. And I think if even a fraction of those come to fruition, it will be fantastic. And hopefully Master will have played some small part in doing that. So thank you to you, Tanya. Indeed, thank you to all of the different chairs, all of the different speakers, the presenters, the panelists, more broadly, the different advisory panel members who contributed today, but over the last 4.5 years, all of the individual researchers, experienced and initially not so experienced, but even the PhD students that started with us are now the next generation and have really, it's been fantastic to see their maturation and the fantastic work that they've done during the course of the project. I'd like to particularly thank Mairead Coakley. So anybody that was involved in the master project over the last 4.5 years knows how much Mairead, as the project manager here at Teagasc, did to make sure the show was kept on the road, and it really wouldn't have happened if you were relying on me rather than her. So a big huge thanks to Mairead. Also thanks to Mairead for doing the background work for today's series of presentations and setting it up. And a big thank you to Padraig Nolan and Muireann Negan for doing that as well. So thanks to so many different people. Thanks to the EC, the EU for funding. And now I'll wrap up by thanking all of the people who were able to attend here. I hope you enjoyed it and learned a little bit more about microbiomes and are inspired to continue to work in microbiomes, study microbiomes in the future, or just employ microbiome-based tools when they come to you in your supermarket or on your farm or wherever else that might be in the future. So thanks, everybody. It's been a great pleasure.